The next speaker, Dr. Vashista, he's going to talk about the nephrology projects.
Thank you. Yes. Good afternoon, everybody. My name is Himanshu Vashista. I'm a staff scientist at the Department of Nephrology. And we have several research going on in the Department of Nephrology, mostly on the basic science side. So the first project that's going on, the role of P66 in the diabetic nephropathy. A little bit introduction on the P66 is an adaptive protein that is directly involved in the ROS metabolism of mitochondria. What it does during the stress gene activation, it moves to the mitochondrial wall and helps generation of one extra molecule of oxygen. And that extra molecule of oxygen that reacts with the water and creates a molecule of hydrogen peroxide that initiates the permeability transition pore opening of mitochondria and swelling of the mitochondria and the mitochondrial death happens. So in this protocol, we have studies in the several cell lines that if we remove this particular gene, then the cells are rescued from the apoptosis. And in vitro studies that we did on the various renal cells as well as into cardiomyocytes. Then further going and to validate this hypothesis in the mouse models, we created, we took the Akita mouse model, which is diabetic, and you can see the glomerular sclerosis in the middle panel, which is Akita and has a typical pathogenesis. On the other hand, this mouse model actually is insulin mutated mouse model that has hyperglycemia at the age of 6 weeks only, but hyperglycemia goes as they grow in the age, and at the time of around 12 months of age, they have approximately 700 milligrams per deciliter of the hyperglycemia, and that's where we see the pathogenesis in diabetic nephropathy. When we created the mouse model, we deleted the gene P66 from this mouse model. We found out the rescue in this system. So when we did this electron microscopy studies, on the left panel you see the intact podocyte. Podocytes are the cells, probably most of you know podocytes are the cells that are that do not regenerate after the birth. So if we can make them resistant to the stress gene program, then we can just rescue them. So that's why, because the P66 gene is actually it's a longevity gene, and removing that elongated the lifespan about 33% in the mouse model. So we created that and found out in the right panel you see that there is a less effacement and the podocytes are intact. When we moved further in this protocol, we did found that in the P66 knockout Akita also we found some of the cellular apoptosis, but kidneys were functioning well in comparison to the Akita. So we thought that there are some cellular turnover which is going on in the kidneys of these mice. And finally, we isolated and the MSCs from this mouse model and planning to inject into the kidneys of the diabetic mouse models to see if they regenerate or they participate in the renal regeneration. But the major challenge that we had is after the transplant how to track those cells, because it was not that easy. So we converted this mouse to the green fluorescence protein expressing mouse completely, where every cell of this mouse has green fluorescence in it except the hairs. So now we have isolated the MSCs from this mouse model and characterized, and we will be transplanting these mice, diabetic mice kidneys, and to see if they show some renal regeneration in those. So another project that we are having, the second project that we have in nephrology research is the role of proteasome inhibitors in membranous nephropathy. The proteasomes are associated with the pathophysiology of the progression of membranous nephropathy. The nuclear transcription factor is a crucial transcription factor that regulates the expression of wide variety of inflammatory genes, including the cytochrome P450. The proteasome degradation degrades the I-kappa-B, which results in the nuclear translocation into an F-kappa-B. And cytochrome P450 plays a major role in the puromycin or pan-induced glomerular injury that mimics the MCD or minimal chain disease in the humans. So in order to perform this study, we are taking two proteasome inhibitor. One is the traditional MG132, and the second one is the carfilzomib. Carfilzomib is a proteasome inhibitor that is being used currently in multiple myeloma patients. But over there, we are trying to induce the apoptosis in the cancer cells. But here, we are taking just 1/5 of the dose, and we found out pretreatment of these rats and induction of the membranous nephropathy through the PAN actually rescue the kidney from the PAN-induced nephropathy models at 10 days only. Another project that we have is ongoing studies in the basic science of reversal of epigenetic changes in photocytopathies. So gene expression is actually the outcome of chromatin modifications, and that can be reversed with the chemical agents. Losartan has been used in the proteinuria patients over the past 3 decades, but there has not been any new drug came into the market. So we try to find out if combination of losartan with certain other drugs like hydralazine, which is a demethylating agent at the dose of 1/10, which is normally used in the human. I mean, this is a hypertensive drug as a normal dose, but if we use this 1/10, then it works as a demethylating agent in epigenetics. So in Akita mouse model, which is again a diabetic mouse model, we see the VDR, which is vitamin D receptor, is downregulated, and snail transcription factor that is directly associated, or that binds to the transcription factor in E-cadherin gene is upregulated. When we treat with the losartan, which is the drug for the proteinuria, then we see the partial rescue in those kidneys. But on the other hand, just a Akita mouse model that was treated with the hydralazine shows pretty much same result. But on the combination, the rightmost panel, you see the total rescue or more than 90% rescue from the kidney pathology. This was on the basic science research side. Another protocol just came in. Dr. Velez recently joined us, and in collaboration with Dr. Namar at Tulane, he is running this protocol in which aminopeptidase A and intrarenal renin-angiotensin system balance in the glomerular disease he is evaluating. So this project just started. Apart from the basic science research, we have several clinical research going on in the area of glomerular diseases, anemia of CKD, and we have acute kidney injury, chronic kidney disease, and we are working, and these are in the planning stage of the acute kidney injury investigator-initiated protocols, and the acute kidney injury and still end-stage renal disease programs. Any questions so far? So these protocols are mostly investigated, before that, these all protocols are sponsor-initiated protocols where the recruitment, they're in the various stages, submission or recruitment or enrolling the patients, where there is a potential for you guys to involve, and also in the basic science research, because we are planning to take all those basic science research protocols from bench to bedside, and we'll be trying to evaluate those hypotheses in the patient samples very soon. So, if you think, Dr. Garcia, I think you have my contact information, and I can be contacted if you have any question for this. Well, thank you so much for listening.